Welcome to calculating matrix effect in a spreadsheet session. First of all, we are going to see how to calculate matrix effect based on the analyzed signals. We will see it on the example of methiocarb sulfoxide determination in tomato matrix. To calculate matrix effect based on analyzed signal, we need to have two kinds of samples. We need to have the blank extracts after sample preparation, which have been spiked with analyte in a known concentration. And these samples have been measured and we have an analyte signal. Also, we need to have the analyte signal in the solvent. And this measurement of analyte signal needs to be carried out on exactly the same concentration level as the analyte was spiked into the blank extract. So basically we need to have two types of analyte signals, one's in the post-extraction spiked matrix and the other in the solvent, and both at the same concentration level. So here for methiocarb sulfoxide analysis in tomato matrix, we have two types of data. We have the post-extraction spiked results for methiocarb at 0.4 milligrams per kilogram concentration level. There have been 3 measurements and the peak areas, so-called signals, for the analyte have been recorded. Also, the analyst has carried out the calibration graph in solvent containing 6 data points. Out of 1 is also at the same concentration level as was done the spiking. So if we want to do the calculation for matrix effect based on the signals, it's very important that we have exactly the same concentration measured for the in solvent and in the blank extract. So now to calculate matrix effect, we have 3 data points in the blank matrix that has been spiked after extraction. So to do calculations, we first need to average these 3 signals, and then we can proceed with the matrix effect calculation. So first we are going to average these signals. Now when we have the average signal, we can compare it with the analyte signal in the solvent at the same concentration level, or to say we can divide it. So from this we obtain the matrix effect. We divide the analyte signal at measured at 0.4 milligrams per kilogram with the analyte signal in the calibration graph at the same concentration level. And now we obtain it, this matrix effect in the percentage scale when we multiply it with 100% also. So we have in this case obtained the matrix effect of 28.1%, meaning that as 100% denoted no suppression or enhancement, values above 100% denoted ionization enhancement, and values below 100% showed us ionization suppression, that in this case we have a very strong ionization suppression. Almost 3/4 of the signal that is present in the calibration graph is suppressed by the tomato matrix for methiocarb sulfoxide.